I just want to start by saying thanks for having me. I'm excited to talk to you all today about ImageJ. And just again, I know Chris sent you the link to the presentation, the slides. They're kind of helpful to have as a resource. And I just wanted to let you know that they actually will continue to live and be accessible via the so at imagej.net/presentations, you'll see them listed here, CZI workshops, and under the workshop section. So just so you know, and so you can follow along. So I'm just going to start. Okay. So my name is Ellen Dobson. I am an assistant scientist at the University of Wisconsin-Madison, working in the laboratory of Kevin Ellisiri. I've been there now for a few years, and I'm a part of the ImageJ team. And this is a part of what I do, is doing these training workshops with new users, as well as advanced users. So today I just wanted to, we're going to go over basically an introduction for ImageJ, but I actually wanted to start with an announcement that ImageJ is actually part of the new Center for Open Bioimage Analysis. That's called COBA. This was established in 2020 with a P41 grant through the NIH to co-PIs Ann Carpenter of the Broad Institute and the Cell Profiler Group, as well as my boss, Kevin Ellisiri at UW-Madison, and we're part of the ImageJ team. So we're basically bringing together Cell Profiler and ImageJ as a national biomedical technology research resource. And our purpose is to develop and maintain novel technologies throughout the biomedical research community. So I'm just mentioning this because there's a lot of resources here. If you go to openbioimageanalysis.org, we are also looking for collaborations. So if you have any bioimage analysis needs or wants, you can connect with us there. So I just wanted to put that out there. So again, we're going to start with just some introduction to ImageJ, really just getting started with the ImageJ user application. So what is ImageJ? Well, ImageJ is an open source software package specifically built for scientific image analysis. It's not Photoshop. This is really an end user application for doing science. It's also a reusable set of libraries and a shared framework for image analysis. So ImageJ is really built on many people's research, and it's extensible, right? So many people can contribute to this project, and science is the same. So as well as it's the fact that it's open source, right? So it's all about reproducibility and transparency. So today we're actually going to focus on the end user application side of things, and therefore we'll employ the Fiji distribution of this software. So saying that there's three main flavors of ImageJ. We have the original ImageJ here. Then we have ImageJ2, which is basically a rewrite of ImageJ for multidimensional image data with a focus on scientific imaging. There's Fiji, which is what we'll focus on today. And this is a distribution of ImageJ2 that comes with a whole host of plugins and tools for scientific image analysis. And it also bundles the original ImageJ1. So the original ImageJ, so the ImageJ version 1.x is the stable original version of ImageJ that has been under continuous development since 1997 by Wayne Rasband. This was developed as a cross-platform version of NIH Image, which is what the original software platform was. And why is this important? What does this all matter? It's been around for a very long time, right? Well, it does matter because our group at Madison basically rebuilt the architecture of ImageJ under the hood to bring it up to date, and that's our ImageJ2. For example, ImageJ1 can only handle 5-dimensional data sets, and now with ImageJ2, with the updates, we can handle multidimensional, newer imaging modalities like light sheet microscopes and so on. So ImageJ Fiji, so ImageJ2 and Fiji basically have a legacy layer that keeps things backwards compatible. So any macro or any sort of function that worked in ImageJ1 still works in the current versions of Fiji and ImageJ2. So the aim was to really keep the community, because that's the strength of ImageJ, is keep the community strong and cohesive as possible. So again, everything's backwards compatible. So Fiji. So Fiji is, again, it's a distribution of ImageJ. It really is just ImageJ. That's what it stands for, Fiji. It's essentially just a collection of image analysis tools and plugins that was made specifically for life scientists. So everything we discuss in this workshop and the next week's workshop is in the context of this distribution of ImageJ. And Chris, you can download and have the most updated version of Fiji on your systems if you want to play along with what we're doing in the workshops. And again, it has a legacy layer. So everything's backwards compatible. And really it's really an easy to install and updated tool. So that's why we recommend new users to use it. It's easy to install, easy to update. It bundles tons of plugins and tools, and there's tons of documentation about this particular tool that are available to us online. So what can Fiji do? What can we do with it? These are a bunch of the types and tools and methods that one can carry out in Fiji. So in general, we can open and visualize multi-dimensional data sets, including 3D volume rendering. You can do registration, stitching, segmentation, tracking, image measurements, the list goes on. And we're going to get a taste of that today and next week. It's really the go-to place to learn for beginners as well as more advanced users and developers as well. This site has actually been very recently updated, and it's now hosted and can be edited via GitHub. And we've spent a lot of time curating the information in here, but nothing's perfect. And actually, we really lean on the community to edit pages and ask for better content as well. So yeah, if anything you think is missing or whatever, please reach out to us, and we love to just continue to improve this resource for the community. So saying that, this is probably the most important takeaway of today's workshop, and it is sharing with you all the Scientific Community Image Forum. If you haven't heard of it yet, this is like the go-to place for all your image analysis needs, pretty much. If you want to save time, right? But avoid pitfalls and do good science. This is basically the whole open source community has access to this forum, and many members are active on this forum, from users to developers to bioimage analysts such as me. There's a whole host of these experts that are ready and willing to help you with questions. So even if you have extremely basic questions, like I forgot what's the best way of opening an image in ImageJ or whatever, like we're here to help answer those questions, right? And it's a great tool to search. If there's something that you're trying to use a new plugin or tool, you can search and see what other people have tried or had problems with, and see if you can get your questions answered already just by searching on the forum. But this is really the place to ask for help. It's not just an ImageJ forum. It basically is home to a whole host of open source platforms that are specific for all sorts of different techniques and have different tools. So even if you might have a specific question about Fiji or ImageJ, but maybe you just have a general question, like I want to start doing this type of analysis, like skeletonization, and like I want to count branches on this, and what is the best tool to do that? It might not necessarily be ImageJ. Maybe there's another tool out there that's better for your particular data set. So this is just a great place to ask beginning questions when you're setting up workflows. Okay, so let's just go to the software itself then. So let's start with the search bar. So this is like the other main takeaway from this workshop. The two main things are, one, the forum, two is the search bar. The search bar is your friend, we're a feature. And the quick way to get to it is the letter L. So that's like the shortcut to get to the search bar. So I will, sorry. I just want to switch over to Fiji itself so you guys can see it here. So if I just hit L, I go straight to the search bar. You can see I'm blinking in there. And what's great about the search bar is that it not only finds commands, for example, let's say threshold I want to do a threshold. It'll find the different commands, but it also searches for different functions. You can also search on the wiki or the forum for posts. So this is really a handy spot to go to, but you can also, what I use it for is to actually run functions. So for example, I'm going to open the example image blobs. So pretty much everything I do, it's a little slow to connect. But yeah, so everything I do, I run through the search bar. So if I just tap L again, and I want to, let's say, run a Gaussian blur on it, just a regular Sigma. So I can run everything directly from the search bar by just hitting enter. So typing in enter, and you'll see from the drop-down menus, it's a lot of times it's really hard to remember, okay, where does that command live? Like I can't remember. And as long as you know the general term for it, you can just type it in the search bar, find it, and then click enter and just run it right away, which is awesome. So I'm just going to close. Okay. Close all the images there and go back. So yeah, so that's the search bar. And then the main window. Okay, it's not the fanciest of software platforms as far as its aesthetics. And we know it's really not that remarkable looking. And we held on to the original design from, but there's a lot going on under the hood. This is a very powerful software. It just doesn't like to show it all. It's not a very showy-offy kind of platform. So there's 4 main segments, right? We talked about the search bar that we already showed. And then we have the menu. So again, those are all the goodies that are hidden inside ImageJ, but we're going to lean on the search bar in order to access most of those goodies. So you don't have to literally click on everything. That's kind of annoying. You can work much faster by just typing. Then we have the status bar. So what's cool about the status bar is it actually, it sets as a memory reset. That's just like a side thing. If you click on it, it just resets the memory. But also, if you click on it, what's nice too is it can also show you what version of ImageJ you're running. So let's say you post on the forum, oh, I'm running into this error. I don't know what's going on. Most of the time, people ask you what version of J, and all that information can be found just clicking on the status bar. And then again, yeah, we have the list of tools. That's the other part of the menu bar. So drawing different regions of interest and things like that. So you'll see some of these guys have little red arrows on the right-hand side. That means there's other options that exist there. So if you click on the straight line tool and you right click on it, you'll see that you can actually change it to example for a segmented line, a freehand line, or an arrow. So that's what those little red triangles mean. And then also many of them, if you just double click them, you can also change some more control settings. So for the line width, if you double click on it, you can adjust that as well. Those are little hidden goodies that aren't so obvious. So then, okay, what's great about, again, I mentioned it before, is staying up to date. So Fiji is built on the newest version of ImageJ. And the updater is really a key feature here. So it tells you when updates occur. Previously, in the original ImageJ version, ImageJ 1, you were in charge of installing plugins and tools and extensions, and then also checking to see if they needed updates, if there were bug fixes or anything like that. Now that responsibility no longer rests on your shoulders. So you can lean on this feature as the updater. So when bugs are fixed or updates are implemented, it just does it immediately. It automatically downloads everything. So if you go to the help dropdown, you'll see there's actually 2 updates listed here. There's the update ... which is the update for Fiji. And then there's another one that's a little more specific, update ImageJ. So that one's actually for the ImageJ 1 version, the original version, sync completely. So usually for the Fiji updates, we wait till there's a few more changes before we release certain updates. Whereas the ImageJ 1, Wayne Rasband, the original programmer, the developer of that tool, still does bug fixes and stuff. So sometimes he'll do bug fixes before the big release. So there's occasionally times, very rarely, but sometimes where, if you post on the forum and say, okay, this command has got a bug in it, Wayne fixes it, and now there's a new version of ImageJ 1, you can actually just update that through the update ImageJ, okay, before you have to update before there's a full update for Fiji. Also, when you run this, when you run the updater, which I'll just quick do, you see this menu pop up, and all the updates that, so it's searching for updates. It'll have a list of updates that will be installed on your system. In this first, I think it's because of Zoom and my internet. Yeah, okay. Normally there would be like a list here showing like which plugins and tools are going to get updates because of bug fixes or whatever. But then you also have this option of manage update sites. So an update site is essentially a web space used by the updater, which enables users to share their macros, scripts, and plugins with other researchers. So there's a huge list. If you click on that, there's a huge list of all these developers around the world who have built tools and want to share them with you. Okay. So you can see like a lot of them I don't even have installed. There's a lot, and there's more and more every day. I'm constantly building update sites for people. But there's tons of great tools and plugins that you can just enable. So that's what, when you click on this check mark, it's called enabling an update site. So you'll hear that language on the forum and such. And that gives you access to those tools without having to install or anything like on your, okay. I would recommend, for example, there's a couple on here. BioVoxel is an amazing tool for image analysis. The developer is active on the forum, and he's built tons of extensions of built-in ImageJ commands, but then he extended them to have even more functionality. That's a great tool. Clear Volume is a new 3D renderer where you can actually do 3D rendering during acquisitions. So you can actually see like a rendering occurring as you're doing the acquisition. And then another one that if you've got this open and later on in the workshop, we're going to need this one enabled, this image science. So just file that away if you don't want to do it now, but just make a note that you should enable this image science updater because we're going to use that later for a machine learning plugin later. We'll need that. So then, yeah, you just can apply those changes and off you go. So that's updating ImageJ. Start with just the basics. So like opening data, the first step, right? What is the best way to open data? Well, as you are all well aware, I'm sure, there are dozens and dozens of file formats for image data, right? The scientific imaging field has well over 150 different proprietary file formats, right? Every single vendor has its own. Leica's is file formatting, and the way they save metadata of your images is different than Zeiss, which is different than Nikon, and so on and so forth. So this obviously presents a huge problem when you're trying just to open your images, the first step. So ImageJ does its best really to open images the best it can, but it's not always perfect. So there's two ways to do it. There's one, the quick and dirty way, which can be very dirty, and two is using bioformats. So we're gonna I'll quick show you how to do the easy drag and drop. So for example, I have an image here. I just drag and drop it here, and it opens the image. Okay, that's great. But sometimes it doesn't read in dimensionalities correctly. Sometimes you can see like, oh, maybe it didn't calibrate correctly because ImageJ is just trying its hardest, but it's not very good at it. So the tool we recommend using is called bioformats. So if you type in bioformats, we're going to import that same image using bioformats, and I'll just show you the window here. So bioformats is essentially a library for opening various file formats. Their job is literally to say, okay, hey Leica, hey Zeiss, we want to know how you save your metadata so we can read it incorrectly. And that's what bioformats does. So if you're dealing with any sort of quantitative analysis or anything, use bioformats to open your images, 100%. If you just want to come around with something, drag and drop, it's fine. But if you're developing a workflow for your image analysis stuff, please use bioformats. It's the best way of reading in your data. So there's different tools and options on here. What's great about it is that, as you can see, the information explains what each of these things mean. You can open and display the metadata itself. So if you want to read that, that's a handy thing to enable. You can also use virtual stack, for example, if you have working with really big data sets and you just want to have a quick view. It only reads one image plane into memory at a time. But the thing about this is that, yeah, you're not looking at the actual data. So if you make any adjustments of brightness and contrast and things like that, and you shift to the next plane, all that is lost, right, that you previously did. But it's good for really large data sets. You can open it into memory. Do the same with crop on imports. If you have a large, for example, like a large H&E histo slide scan you did, but you just want to zoom on one particular region, you can crop it on import in order to open it. So again, that's bioformats and opening files. Okay, so then we have the image window. So there's a lot of information that ImageJ gives us when we are just looking at our images. So we're going to go ahead and open this mitosis example. So if you open that, so you've got different things like you see the title of the image, you see the zoom factor. Up above, you'll also see the data set position. So which channel you're in, which Z slice, which time point. You can see the image size. So whether it's in pixels, it always gives you the X and Y dimensions, but then if your image is calibrated, it will give you those real measurements as well. So then you know your image is calibrated. So you can also check and see if it's calibrated correctly. And then the image type, 16-bit, so on and so forth. And then the different positions and channels and such. So again, if you open that mitosis image example, it might take a little while. My internet is just a little bit slow at home. Sorry, guys. Yeah, so again, so right now there's no zoom factor shown because I didn't zoom, but you can actually zoom quite easily over an image if you move your cursor and just hit the plus or minus buttons. That's like the quickest way of zooming in. So if you move the cursor, it'll zoom into wherever the cursor is. And then you can see the zoom factor above. And also you can see when I move my cursor on the status bar of the main window, you can see what position I'm located of my pixel and then the value of that particular pixel. And this, obviously we're in this first channel. Now we're in the second channel. You can see it turns to green and we're in channel 2, and then the values would change according to that channel. Okay, so pixel types. So bits, I'm sure many of you already know this, but it's a good idea to just go over it again. So bits refer to the number of possible values that are available for each pixel in your image. So each pixel is a measurement, right? That's what we're doing. We're acquiring measurements when we're doing imaging. And we need to decide on a precision for that measurement. So the more precision it takes up, with the more precision, it takes up more space in memory, right? So it's a larger image file, the more bit depth, the higher bit depth you go. So these are the main types of images that ImageJ deals with. So 8-bit images, which is 2 to the 8th different values. So 256 levels, value 0, 16-bit. So that's 2 to the 16. And then 32-bit, which is called a floating point number. So this refers essentially to real numbers. So essentially it's scientific notation with limited numbers after the decimal point. It also takes into account positive and negative values. So if you're subtracting, for example, if you're subtracting one image from another, it's a good idea to convert it to a 32-bit. So you don't clip your pixel values off at 0 as you would with an 8 or 16-bit image. And as a side note, if you're doing any sort of quantitative analysis, you should always be saving your data at least as 16-bit images, minimally 12-bit images. 8-bit images are really just for qualitative analysis and looking. If you want to do anything quantitative, it should be minimally 12-bit, ideally 16-bit images saved. And on the side note, never, ever, ever save your images as JPEGs. I hope you all know that. And if you don't, that's okay. But please don't ever save your images as JPEGs. JPEGs is not designed for quantitative image analysis. It's just made for looking at pretty pictures that you take of sunsets and things like that. JPEGs actually introduce a lot of artifacts and things, and basically ruins your data. So save them as TIFFs, or ideally the original proprietary file format, and then you can just open them with file formats. Okay, so we'll move on to lookup tables. So lookup tables, or LUTs, like most people refer to them as LUTs, lookup tables are made up of intensity values, or images, as you know, are made up of intensity values, sorry, and they're numbered, right? And these values can basically be assigned any color. It's the color part is arbitrary. As long as you know that. A LUT is essentially a table that assigns possible pixel values to particular colors that are then used to display them. So it's for display purposes only. This doesn't change the value of your pixels. It's just so you can see things more clearly. So these are a few of the LUTs that I find more helpful. So ideally, you're looking at your images in grays. So our eyes actually can distinguish differences in gray levels most effectively. So usually when I'm looking at images, I look at it in gray levels. High-low is also really nice. So it gives you in gray levels, but then also so you can see over-saturated pixels, which are in red, and under-saturated pixels, which are in blue. So that's also nice to see whether you're over or under saturating your images. And royal is also a nice one. It's good for dynamics and colors, but ideally grays or high-low is. Sorry, I got an itch in my throat. I keep coughing. Okay, so getting to know your data. So again, there are better ways to visualize your data besides just looking at the images themselves, right? Our eyes can deceive us. So as you know, images are numbers and measurements. So therefore we can use a histogram and actually look at the numbers of our images. Excuse me. So we'll open the example boats. And then again, so, this was also a little thing that I should have told you. No, you can see how my boats image kind of hid the main window. If you just hit enter, so no matter how many windows pop open, that's one thing about Fiji, you get all these little extraneous windows that open. If you just hit enter, that main window automatically pops forward. So that's just a nice little secret thing. So we'll just type in histogram. That guy. Oh, what are the pixel values in my image? Excuse me. I have an absolute tickle in my throat. So if you move the cursor over the histogram, you can actually see how many pixels have that particular value in this image, and you can actually list them out. So you can see quite easily, right, in the list that you're not over or under saturating this particular image. Which is obviously what we want to avoid. So are there any, I see some chats. I just see that there's things popping up in the chats. Are there any questions yet?
There's an installation issue that I'm chatting with someone about.
Okay. Do you need any assistance, or is it good?
I think I found a solution, but maybe
okay.
I can ask real quick just in case.
Yeah.
On a Mac, someone is having an error saying that a Fiji is a read-only, so they can't update anything.
That is weird. Is it your own computer, or is it like a university computer or something where you don't have access?
It seems like
because you need access to download something off the internet.
No, it is a work computer, but I do have the administrator privileges, so I am allowed to download, and I don't have difficulty downloading other softwares. But this one I have tried, like, a few times to uninstall and do it again. But every time I do this on my computer, it says it's read only.
Read only. Okay, so this would be a good that without looking at your system and stuff, this would be something that would be hard for me to quick answer on the fly. But if this continues to be an issue, we can connect offline, well, online, but not here.
Okay, thank you so much.
And try to get that. I'm sorry, it's not a very common issue. Is there anything else? I'm sorry, I have this tickle in my throat. I keep on coughing. I don't have COVID.
No
I just have like a tickle in my throat, and I'm drinking all my water. I'm panicking because it's going to be gone soon. Okay. So let's just keep going. So another way to look at your data sets is using plot profiles. So we're going to open the sample image called blobs. Again, we're going to use the line tool to draw a line across some of those blobs and then plot that profile. This is kind of just a way of showing you that it's good to look at the numbers, the underlying numbers in your images. So we're going to open that blobs image. You can do this with me and take the straight line tool and just draw it across like one or two of those blobs. Then we'll go to the search bar. So if you just want to hit L or Control L if you're on, I'm going to plot profile. So you'll see something that's strange, right, about this profile in this image. So across, you can see the two dots, right? You can see the two blobs. We have a peak in intensity over those two blobs. But that's a little weird, right, with the image that we're looking at, right? Because it's dark. So the blobs are actually dark. So you would think it would be the opposite, right? That you see a dip in pixel values over the blobs. Well, if you look closely, you'll see that this is actually an inverted LUT. So the higher pixel values are given a dark shade. And then the lower pixel values, if you move your mouse and cursor over and look at the status bar, the lighter or brighter pixels are actually a lower value. So it's the opposite of what you would think. You can actually invert that LUT again. Excuse me. So it makes more sense. So there's actually a LUT drop-down menu that I use a lot because it's just faster. So you can just quick invert it and then it makes sense, right? Then it makes sense with what we normally see, that brighter pixels have higher value than the lower ones. So that's just so that, again, our images are numbers, and all these visualization things with LUTs and all this stuff is just that for visualization. It doesn't change the numbers. So always rely on what the values of your actual image are. You can also plot profiles in Z or in Z, just so you know. And then 2D visualization, okay. So there's a couple tools that we can access in ImageJ for accessing 2D visualization. So we'll open again that sample called mitosis. So if you follow along, we're going to just open mitosis again. And the default in ImageJ is to open everything in red and green, which is not ideal, right, for people with colorblind issues or who are colorblind. So you can actually change the LUT directly and change that to magenta. So if I just type magenta, now it's magenta. But what a nice tool to use is the channels tool. So composite means that we're looking at both channels at once, but you can also switch it. So you're just looking at a single channel at a time. And so color, or in the composite, you can turn off one channel or another. So that's just kind of a handy thing to know, as well as properties, image properties. Yeah, properties. So this also tells you the number of slices, frames, the pixel size. So if you have a calibrated image. So image properties is a great thing to check if something just doesn't seem right in your image and you just want to give it a double check to make sure that that metadata was read incorrectly. So yeah, so again, this is the tool. This 2D visualization tool is one that is built in with ImageJ, part of the original ImageJ. So it's limited to 5 dimensions, right? So X, Y, channel, Z, and time. So that's the most dimensionality image the original ImageJ can open. But there are other tools now, again, available through Fiji, where you can look at images with arbitrary dimensions. And one of those tools is called Big Data Viewer. So I'm not going to go over Big Data Viewer today, just because it's a beast of a plugin. It's amazing. It's getting into it, but once you're in it, it's a fantastic tool. So I recommend anybody who's working in higher dimensionality data sets to look at the Big Data Viewer, Big Data Viewer. Okay, brightness and contrast. So this is sometimes you just want to be able to adjust just for visualization purposes. So if you open boats and then you type in brightness and contrast, which we can quickly do, that boats example, and then brightness and contrast. Oh, I can't access this right second. Okay. You can actually adjust these pixels, right? But again, this is just for visualization. You're not adjusting the pixel values at all. The only thing is, there's one button in this thing that will actually change your pixel values, and that's if you click apply. That will actually pixels that are displayed and then clip off your data. So you never want to click apply because that will rewrite over your pixels. But everything else on here, this auto, all it's doing is just adjusting visualization purposes. I mention this because a lot of times users open images for the first time in ImageJ and they're just totally black. And they panic because they're like, oh my God, my image is gone, my data is gone, it's corrupted or something happened. But 9 times out of 10, it's just ImageJ is opening it and it's just not visualizing it correctly. So the first thing you do is if you open an image and it's black, is open brightness and contrast and just click auto. And usually it'll just readjust how it's displayed. It's only a display issue, the data is still fine. And you can again always move your cursor over the image and check out the pixel values inside the status bar to make sure, okay, no, the pixels are actually there. They're fine.
We
Okay.
have a question about how would you apply the same brightness and contrast across several images?
Yeah, so I don't know if this is macro recordable. It might be, but it's going to adjust automatically based on the individual image. So unless you acquire them all in the same way and stuff, I don't know. Well, one, I don't know in what case would you need that to be done? I mean, because it's really just for visualization. So it has nothing to do with analyzing your images or doing any quantitative measurements.
There's a suggestion about using set to apply to all other images.
Oh, maybe that's a function that, yeah, maybe. It up in the, I would even have to look that up in the ImageJ user guide. So that's actually linked through the ImageJ website. But if you just Google ImageJ user guide, there's an extensive user guide that goes over. So if you type in ImageJ user guide, you search for that, you could actually just type in and look for brightness and contrast tool, and then it'll tell you what every single button and function does on that window. But I think that's correct. That sounds correct. But you wouldn't necessarily need to do that unless you're just worried about, I don't know, for publications or something that you need to save the image in a certain range. But yeah. Okay, we're gonna keep going because I'm worried we might run out of time. So this is probably one of the most common tasks in analyzing images that you'll actually do, right? You want to separate out objects of interest. You want to say, okay, this is my object, this is background, this is my cell, this is something else. So we can use thresholding to actually set that delineation. This is the simplest way of doing it. We're going to go over this a lot in the segmentation part of the workshop, which is the next one. So let's just go ahead and open that sample image blobs. Right, and we're going to just run the thresholder again. So it's threshold ... And you'll see a lot of these functions with a ... at the end. So those are usually like the built-in functions of ImageJ. The common ones that are with the original ImageJ, the ones that we're using today, they're usually like something ... So just, so that's, and here we have our thresholder, right? We don't have a dark background in this case because it's inverted. So you can actually manually, move these sliders and pick the object you want, right? Like, okay, this is covering my object well or not. But you can obviously tell that is kind of an issue. You don't want to do that. You really don't want to manually slide the slider, especially if you're analyzing a couple dozens, if not hundreds of images. You can't manually threshold each one and justify it, right? Like you have to have a more objective way of setting thresholds. This is introducing a lot of bias. If you say, okay, well, I'm going to set it here for this image and set it somewhere else for another image, you can't really, those are apples and oranges. So what we want, ideally what we want to do is use auto thresholding methods. So clicking this auto button, it's using this default algorithm in order to find a threshold. But there's tons that are available and out there that work for different types of objects, different types of images, good or bad. For example, like Atsu is like the go-to one for round, good signal to noise type images. So it's really good for like DAPI staining for isolating nuclei and stuff. Atsu is good. The default actually of ImageJ is based on Atsu. But you're like, okay, so which auto threshold do I choose? Like, how do I even know? There's a whole menu. I have to go through each one and like look at them. Well, we have this tool called auto threshold that will, so we have black objects on a black background. So I'll uncheck that. We have this tool called auto threshold, which will run the threshold on, or the auto threshold for each one of those algorithms. So if you zoom in, you'll see that, okay, that one's for mean. This percentile one doesn't look to, Atsu looks pretty good. So then you can get a quick idea of, okay, this one looks good for the objects that I'm interested in thresholding. So yeah. The other thing I would say is that, yeah, auto threshold is great because it will automatically do that cutoff for you based on the distribution of intensities in your images. But of course, note is if that distribution changes, then the threshold's going to change too, which is good and bad. Good if that's what you're, it's good and bad. So for example, you can bias your measurements if you're using the same signal for detecting an area as you are for measuring the intensities, right? So it's kind of just like having an Acton lane in your Western blots. You need something that does not change in your data to accurately measure the other signal that is changing. So setting thresholds on the same channel as those you wish to measure, the variations you could observe. So it's going to affect your outcome, your conclusions. So if that makes sense. So ideally have a second channel that's like steady. So for example, if you're looking at a nuclei marker and you have it stained with a fluorescent label, then use DAPI to do your segmentation and find your nuclei. And then you can measure and segment your object, and then you can measure that other signal within, for example. Okay. Regions of interest. So this is something that's like usually follows thresholding, and we're going to go through this again, drawing regions of interest automatically in the next workshop. But I'm just want to introduce this stuff to you, and we'll just do it kind of a quick and dirty way with manually. So we're going to open this clown image, and actually we're going to duplicate him right away. Oops, sorry. Duplicate again. So we're going to have two creepy clowns that are open. Then we're going to take this freehand selection, which is this bean tool, this Roy tool, and I want you to try and draw his nose like around. We're going to try and measure like the area of his nose. So I'm not very good at this, but if you click on it and then you start dragging, and as soon as you let go, it closes the object for that freehand tool. So we'll just I'm terrible. Draw that circle around. Okay. And let's say I want to measure that, right? So I want to actually measure, sorry, some different aspects of that nose, like the area, the mean intensity perimeter. So I'm going to print out, if I click measure or type out measure in the search bar, or if you just hit M, Control M, the shortcut M again, so it just measures the same tool or the same region. Again, you can actually adjust what is calculated. It's called set measurements. So set measurements will basically is a list of, sorry, some of the Zoom stuff is in my way, so I can't click and drag this, but the set measurements basically is a list of all the different things that can be calculated in your images when you have regions of interest. So you can select or deselect whichever ones you want to measure, which is why my print off might look a little different from yours based on this. But let's say, okay, I want that measurement, but now I want to get the same exact measurement, right, in this clown. So I want you to try and draw the same exact region of interest in the second clown. So I want the same area, same perimeter, like everything is the same. Well, obviously that's impossible when you're trying to do it manually. For regions of interest, right? Ideally, you want to draw them without doing it manually, which is what we'll get to next in the next workshop. But let's just, we want to save our ROI, right? That's the point here. So if you use the magical letter T, so Control T or just hit T in a Mac, it'll pop up this ROI manager and it will save that ROI. So it hasn't been saving it super well lately. So it should pop up this ROI manager and save. If you hit T, then you should save this object here. We can actually overlay that on the second clown by toggling show all. And see, now we've overlaid him here. So if we click measure again, we're going to actually end up getting the same exact measurements that we did before. Okay, so that's a way that we can use ROI manager is going to be your new best. Oops, sorry, is you're going to be your new best friend. And that's how we save ROI and be able to see how we can automatically generate ROIs based on auto thresholds. So we're not introducing any bias into our measurements and then access them through there. Okay, so I'm just going to, yep.
Is there a way to, if you're drawing a circle, to kind of undo the last few steps that you drew or something? So if someone's drawing
like stepwise? Yeah, that's a good question. No. As far as I know, ImageJ is not that fancy. No. You would have to just redraw it. Yeah. I think like with this guy, you can adjust like individuals, like when you have those little white dots, you know, you can adjust those individually. But with the bean tool and the freehand tool, no. Or maybe manually check them. Yeah, no, it's not so straightforward in ImageJ, unfortunately. Yeah.
Also with the region of interest, can you save that and apply it across analysis sessions?
Yeah, so that's the cool part about, yeah, sorry, I didn't really, in the ROI manager, you can actually save them. So you can save all your ROIs. So when we do the segmentation in the next workshop, we're going to generate a bunch of regions of interest of nuclei, for example. And usually when I'm doing quantitative analysis, I save all of my ROIs for each individual image that I'm analyzing, because you always want, you know, if you need to go back for some reason, to double check, okay, did they look good and what, you know, or you need to access them again. You can save all your ROIs in a zip. It saves as a zip file that you can then again open here in this drop down. So it's in the more section. You can open them again and again, we'll go over this way more detail in the next workshop. This is just kind of a brief introduction of like, what are these tools and things. We're going to do some more serious, like, how can I actually use them and apply them in my image analysis workflows next week? And then how to actually automate them as well, doing scripting. So that's where all the really goody stuff comes in. So yeah, so we did this. Okay, so then plugins. So I've been using this term plugins a lot. I haven't really defined it yet, but essentially a plugin is just a tool. It's an extension of ImageJ. So basically it's a special purpose software component that extends ImageJ's functionality. And that's what's great about ImageJ, right? You've got a huge list of available plugins and tools. Again, we covered that in the manage update sites. All those update sites are based on that let you carry out specific image analysis functions. And I'm just going to introduce you to a few main ones right now for some of the more common image analysis techniques. So just so you have an idea of what's available. Oh, sorry. So one is for colocalization in Fiji. The Fiji plugin that we use for colocalization is called Colo2. So colocalization is just the measurement of the overlap between two different fluorescent labels. So there's also tools there's two different ways of doing colocalization. You have object-oriented methods, and then you have intensity-based methods. So there's tools for object-oriented methods that are available through ImageJ, and then Colo2 is actually for pixel intensity-based methods. So just doing classic measures like Pearson, Manders, Costas, Lee, you get scatter plots, analysis, all those things in your results. Yeah, so this is the tool you would use for colocalization. So registration is also a common method that needs to be applied to images. It's basically the process of transforming different sets of data onto a single coordinate system. So i.e. just lining things up, right? So what are some of the cases where you would need to do registration? Stitching, for example, is just that. It's when you won't combine regions of overlap, right? And you need to find those areas that are in common and overlap them. SPIM data sets. This is when you're imaging large 3D volumes from multiple angles, right? The same object from multiple angles. That's why a lot of researchers embed beads in their samples. They're actually using those beads as a coordinate system that they can then map all the different views onto the same system. And in my case, years ago, like I was imaging on a system that had two confocal, and then the other I could switch to and do FLIM acquisitions, but the two detectors were two different sizes, but I needed to obviously like register them over so I could actually see the same cells, right, in both FLIM and my spinning disk confocal. So there's many different plugins that are available for registration. One is feature extraction. So this guy basically automatically finds points of interest, detects them automatically, and then uses those to register one image onto another. So it'll find points in common and then use those to register. Register virtual stack will register a stack of images using a single slice as a reference. So you choose which one's a reference and then it'll align it, right, in case you have shifting over time or things like that. Track EM2 is a huge plugin. It's amazing. There's a lot of documentation about this plugin as well. It does a whole host of things, but it also does stitching, registration, extraction, as well as global optimization algorithms to do registration. And then there's Beyond Warp, which does 2D image registration, but it's based on elastic deformations. Okay, and then tracking. So tracking is another common thing where you follow objects of interest over time, right? TrackMate is an amazing plugin. A good friend of mine is the developer of this plugin. He's active on the forum. He's got extensive documentation on the ImageJ on imagej.net regarding this tool and how to use it. Basically, it allows you to detect objects of interest, right? So you first detect them, and then the next part in tracking is linking them over time, right, to generate tracks. So in this tool, you can actually do automated or semi-automated tracking. You can also trace cell lineages, so if cell mother and daughter cells, and track those over time as well. Now for 3D visualization, so there are actually a whole host of 3D tools available in ImageJ and Fiji. You can actually follow along with the updates in that link at the bottom of this slide. And ImageJ and Fiji have been notoriously bad when it comes to 3D visualization tools, but that's changing, thankfully, in part to new development and some new tools. And one of them is called SciView. I wanted to highlight here because that's the most promising one. So this uses scenery as a rendering backend, and there's a great deal of new development going on with this plugin, and you can activate or you can enable this update site for SciView and try it out. But again, it's under development, so it's not perfect, but it's getting there. And though, again, the developers are active on the forum. So if you have questions or anything, you can see them there. So we're going to use this last half an hour or so to just what is segmentation and the basics of it. But then we are also going to focus today on a machine learning tool in ImageJ. which is one of my favorite tools. I use it all the time for segmentation. So let's just take a little, does anyone have questions before we move on actually? At this point? No? I'm not opening, I can't see the chat right now, so in the share.
Nothing in the chat. I don't see anybody
Okay.
raising their hands.
Okay, that's fine. We'll just keep going. Yeah, but if you have questions, feel free to also like ask without, like just asking. That's fine too. No problem. Okay, so we'll start off with what is segmentation. So how would you define segmentation? Well, I think the simplest way of defining it would be just object identification, right? I've got objects in my image that I want to say, like, which are the objects that I'm interested in. So how can we identify a nucleus, right? Or a cell wall? How does the computer identify structures from our images? And then isolating these structures. And that's essentially segmentation. It's a partitioning of a digital image into multiple segments. That's like the fancy definition. But more importantly, before you even start workflows and things like that, it's important to think about some common assumptions, requirements needed before you set up a segmentation analysis, to ask yourself some basic questions. And even before you set up your sample acquisitions, right? Your sample prep, it's important to think about these things. So one, do you have overlapping objects? But more importantly, do you care, right? For example, if you're interested in a population signal versus individuals, how your workflow will be developed. If there is significant overlap between two cells, a given pixel can therefore belong to more than one object. And you basically at that point, you can't have individual objects. You can't say, okay, this pixel belongs to this one, not this one. You really have no idea. So that's an important thing to kind of suss out and decide for your biological question, is that important? Another thing is, do you want to count objects or just want to segment regions? Because that will also, you can streamline your protocol and keep it a little bit more simple. For example, if you're counting objects, you can use find maxima, right? To identify objects using the maximum intensity. Though that's not so great if you have really noisy images. It's not the best method. There are other ways to count objects, but that could be one, right? Then you don't really need to say, okay, this is the size of my nuclei if all you want to do is count them, right? Or maybe of pixels, and then you want to know the intensity values within and the ranges and et cetera. And then you need to use more like classic thresholding methods. So then what are the shapes of your objects? So my computer science friend always told me, well, there's three main shapes that biologists are interested in: tubes, blobs, and clouds. So as a biologist myself, like for me, I have to kind of translate that. So a tube would be, for me, in my brain, like a fiber, a dendrite, actin type thing, something that you want to know the orientation of, or the length, or the branching of. Those are the different things you would want to measure of those objects. Blobs would be any circular type object. So nuclei, cells themselves, right? Or organelles, anything that's elliptical, circular on shape. Then maybe you want to know their area, or you want to count them. And then finally, my brain would be like granules or regions of varying intensities, right? And then you want to know that intensity, the range of intensity, the location, things like that. So knowing the structure and shape of your objects will also affect how you build your workflows moving forward. And then finally, what modality are you using? There's a big difference between how you would build up a workflow for bright field than you would for immunofluorescence. And then finally, how does the signal look, right? Do you have a good signal to noise ratio? It's going to be easier for you to segment and less pre-processing steps if you have a good signal to noise ratio. Okay, so we're going to start, actually, there's two main approaches in segmentation. One is a tailor-made and flexible workflow, which is like a classic version, and that we're going to go over in the next workshop. And then there's one that takes advantage of machine learning. It's relatively, we let the computer really do the hard work. So we're going to go over that one right now. So this tool can really solve a ton of your segmentation cases. And this plugin is called Trainable Weka Segmentation. It is fantastic. And the developer of this tool is absolutely amazing and also active on the forum. So basically, it's essentially a plugin that you as a user can train the computer from your input to perform the same task on unknown data later. So it uses powerful tools and classifiers of Weka, which is a collection of machine learning algorithms for data mining tasks. So basically, it combines a collection of machine learning algorithms with a set of selected image features to produce a pixel-based segmentation. So it can say each pixel belongs to a certain class that you define as a user. So stepwise, how does this work? Well, one, you have an input image, right? So you have this image that you're interested in. Then you decide, okay, I'm going to calculate all these different image features. Maybe you'll just do one or two, maybe you'll do them all. And it depends on your image and how complex it is. And then you actually do drawing on your actual image, saying, okay, this pixel belongs to this object or this class, and should get this label, and then this object belongs to that class, which should get another label, and so on. And then what you do is you're training it, right? So then it's a combination of these image features you select, as well as this input labeling, that then generates a classifier that's trained. And then this is what you apply to your larger data set. So then you get a segmentation at the end with your different classes. So this is the main window of the tool. So we're going to actually open it and run it. I just wanted to go over this. There's three main parts. You have the labels on the right side. So you have different classes, they term them. So you can have 1, 2, or more classes. I think minimally you need 2, but you can have more than 2. And then you have the options section here. And so this is where you can choose the different features to calculate. You can load and apply different classifiers, add new classes, and then finally training. So once you have your regions drawn and assigned to different classes and the features chosen, then you can start to train. And it's an iterative process. You will add, subtract, change, the tracings that you draw, add or subtract features you calculate, and then train again until you get a classifier that segments your objects to your liking. And then you can apply the classifier across your whole set of images. So we're actually going to use this image. So if you guys click here, I'm sorry, I'm bouncing out of the whole view. So I clicked, I clicked on that link below. So this is actually an example that was posted to the forum a few years ago now, but it's a fantastic example because someone posted, literally just was like, I have these cell aggregates in micro well plate that I'm trying to segment and I'm having problems. And someone responded, you should use Trainable Weka. And so we actually now use this as an example. So if you just, if you guys right click on it, you can actually download that image, or I think the link is here too, because we'll use that now as an example to open and run. So if you want to do it with me, it's a good idea to have this image downloaded. It is unfortunately a JPEG, which is a huge no-no. So I saved it as a TIFF, even though it doesn't matter, the data is already corrupted. But yeah, so don't ever save your images as JPEGs. But we're gonna open this now then in Fiji and run this example. So again, this person just, and I'm going to do bad drag and drop just for speed purposes. And then if you just type in the search bar, Trainable Weka, it takes a minute to load. Plus my internet's a little bit slow. Yeah, okay. So now you can just zoom in and out like you normally would for an image, hitting the plus and minus. So we have our image here in the window, right? So again, this is someone who has cells, like cell aggregates, they call them, in a microwell plate. So if you just look at the image, right, it seems easy, right? Our eyes are really good at distinguishing what is a cell and what is not. But you can see how it would be hard for a computer to say, okay, this pixel belongs to that, right? You've got these shadows in the wells and the cell aggregates. You also see that you have a bit of background correction that needs to be done. It's a bit darker down here than it is up at the right, right? So yeah, again, we have two classes. We're going to have cell aggregates and then whatever's background, right? So we're first going to click on the settings. I'm just going to move this. I can't move it. Okay, anyway, so the settings, you'll see a whole list. All these top parts are the different features you can calculate. So these are the default ones, I think, that just showed up that are clicked on. So the thing is that it's a very computationally heavy process. So the more features that you calculate, the more basically juice that it's going to take from the computer to run this. So it eats up a lot of memory. So it's always number of features and the number of tracings as well, right? Like we're actually going to draw on the image and draw traces. And if we have like 500 traces or something for one class and 250 for the other one, like that's a lot. So you're asking a lot of the computer because it needs to go through and do all these feature calculations on each one of those pixels. So if you can get good segmentation with the least number of features calculating and the least number of tracings possible, that's ideal. Like you kind of want to minimize that a bit. And then too, you might say, okay, like, well, which feature do I use? Like, how will I know like default maybe isn't working? And in this case, it didn't really work for this particular image. Well, if you go back and look at that forum post, the user who suggested that the person who suggested that they try this tool actually knew what features would be good to use in this particular case. And all of these things are documented as well. So you can read like what are the maths that go behind these feature calculations in your particular situation. But we can actually add variance in this case and structure, as well as changing the maximum sigma. So we're going to open that sigma up a little bit to 32 instead of using the default. And then we can also change the class names, which is nice, or you can actually add classes as well. So we're going to call one aggregates and the other one background, because that makes more sense than class 1 and class 2. Now, in some of your cases, maybe you open this up and you see some of your features are grayed out. That's because you need to enable that update site called Image Science. So if you go back and you're updating ImageJ, go to the Manage Update Sites and enable that Image Science, then you'll get access to all of the features. Okay. So now we're going to just draw. Now what you do like is, oh, sorry, go ahead. Is someone asking a question? No. Okay. Traces and add them, for example, to the aggregate. So you're actually drawing on your image and adding it to a class. So you want to do that across, the entire image, right? It's darker down here, lighter up here. And then also the background. So background traces, including the well, right? And let's just train it based on those few tracings. So you can also change the region of interest. The default is to just draw the squiggly line because that's the minimal number of pixels. If you, for example, want to use a circle or whatever, just know that all of those pixels inside will be used during the computation. So it'll eat up a lot more memory. So that's a trade-off again. So again, you want kind of the minimal pixels. So you'll see there's some here that got on add. You can trace again over there, over that region, right? It picked up some of the well pixels and called an aggregate. Well, no, that's background. And then here again too. And then you can train again. So it's an iterative process. You're going to constantly be going back and forth doing this until you get a segmentation that you think is good. So this one's pretty good, for example. So then you can create a result, which is basically going to be a labeled image that you can then go ahead and segment, which we'll go over those steps in the next workshop. But essentially what you're doing is, I just clicked on get probability. Essentially what you're doing is, in this case, right, you have two classes, right, two labels, the aggregates and background. And what you're doing essentially is the computer is then saying, okay, for every pixel, what is the probability it belongs to one class versus another, which is why there's two channels here. So you're going to see like this one, when I toggle over, it's closer to 1 is the higher probability of it being an aggregate. closer to 0, it means the lower the chance. And so that's why they're like inverted of the other, because of there's just 2 classes. So that's another way you can look at that data. But then once you're happy with that, you can apply it to a larger data set. So it's always a good idea to train on more than a single image of your data set. And you also want to train on representative images on your data set. Now, ideally, you would do all of your acquisitions on the same day with the same sample prep, same exact imaging system, same exact settings, right? And that would basically streamline variations in your data. But sometimes there are a lot of variations in our data. And so you want to make sure that you're training on something that can be broadly applied to all of your images. So that's kind of the big to machine learning. And then this is kind of like a last slide just to have different helpful resources, things that are available to you. Again, the Scientific Community Image Forum is an amazing place. Please go there. I'm there all the time. You can mention me too. I'm ETA Dobson. So if you need help or you're not getting help, I'm on there every day. Then there's the ImageJ user guides, which I mentioned previously. There's a link here that you can access the guides, which are great for all the built-in functions. You can see exactly what every single little feature and button actually does. There's also an excellent presentation on segmentation that's available through here. And then all the workshops and these slides will be available, period. Like, we're all about open source. So these are always be accessed because we want them to be a resource to you down the road. So if something, you missed a little thing that I said, are still here in these slides. But actually that kind of finishes everything. So I don't know, I can stop sharing my screen for the moment and just see if you guys have any questions. If I can help with anything with the remaining time.
So there are a few questions, maybe 3. I'll ask them, I guess, in order of relevance to what you just did. So for the machine learning and training that you just did, can that be applied to an image stack?
Yes, you can apply this to stack images. Yeah. So if you go to the ImageJ website, the imagej.net, and then search for trainable Weka segmentation, there is a whole page of exactly how to use this plugin, what types of images it can be applied to, that gives you kind of a how-to of using this plugin. But yes, you can use it on a stack, yeah.
And then does this machine learning plugin also work on ImageJ, or is it just Fiji supported?
Okay, so yeah, that's that's a hard question to ask, because to answer, one, because are you talking about the original ImageJ version, the 1.x, or are you talking about ImageJ 2? I'm assuming this user is asking about the original ImageJ, and in that case, I don't think so, because this is an actual Fiji plugin. So there's certain plugins that actually are Fiji plugins because they're the ones that automatically come with Fiji when you download it, and TWS is one of those. So it's not like an added on one from the update sites. It's one that's part of the Fiji package.
So even ImageJ 2 might not have it, is that what you're?
Yeah, no, ImageJ 2 does not, because ImageJ 2 is built in the same way as like the original one. The original, this all gets really confusing. The original ImageJ, as well as ImageJ, you have to install and download the individual tools that you want. So that's how that. Some people like that because they can choose which tools that are installed and stuff, but then you have to keep track of how they're updated and if they're updated. Whereas Fiji is just like, you know what, we're just going to give you all these goodies, and then you don't have to worry about installing them individually or updating them as you go. Just take everything. But some people like to pick and choose what they install. But that's why in general, Fiji, it's not like it's a huge software, it's not massive. So that's why we were just like, use Fiji, because it's just so much easier for users.
Right. I want to go back to one before it gets buried a little bit longer, but let's say you have a neuron culture with multiple neuron subtypes, and you want to know the percentage of that. Maybe it might be easier if you read it, I guess, rather than me reading it.
Sorry, I haven't even opened up the chat. neuron culture with multiple neuron subtypes, and you want to know the percentage of a specific neuron type. You can use find maxima to count nuclei. What would you use to count neurons? Oh, given the shape of soma and the neurite sprouting. So it depends on, this depends on many questions, like what is your marker for these neurons? Do you need the entire cell body?
I can explain. Sorry.
Yeah, maybe explain it a little.
Yeah, no, I mean,
this is a type of question that would be awesome to post on the forum. Like if you really, you know, this is exactly the type of question that you can post on the forum and say like, what is the best way of me doing this? Here's an example image, and share an image, right?
Yeah.
But so, yeah, go ahead.
Say you are differentiating neurons towards specific, let's say dopaminergic, but not 100% of the neurons in culture will be dopaminergic. So you have a general neuron, like DAPI staining. So you can use that to count all the neurons in your culture. And then I have dopaminergic specific markers like DH that will mark the cell body and the neurites.
Okay.
So yeah, and I will use that to count how many dopaminergic neurons are. So instead of me personally using the cell count tool and clicking in every single positive, if I was wondering if there's a way like find maxima to count them automatically.
So is there
not like round or anything? Would I need to segment it first, to threshold it first, or I don't know?
Yeah. So do you have overlap? Is there overlap with the nuclei? Like in those cells, is that marker excluded from the nuclei, or
excluded?
Is it excluded?
Yeah.
So that you could do in. Yeah, obviously that makes it trickier, right? Because otherwise you could just say, here are my nuclei, which nuclei are positive for this other marker, and
yes.
Right? Or what you could do is, this is just a suggestion, maybe this isn't the best suggestion, so it's also a good idea to go on the forum to get other people's ideas than just Cole and Dobson. But in my brain, because I've done something similar to this, where you actually delineate the nuclei, but then you expand it by a few pixels, and then you can basically look for that signal inside that donut ring just outside of the nuclei.
So you make like a new ROI based on the nucleus, like
just expand it a bit, so then it's more like a ring shape around your nuclei, and then do that for all the cells. But then you're going to be able to say, okay, the signal exists in this cell, but isn't in this one.
Yeah. Okay.
That's in my brain. That would be the way. It depends on how your images look and how much, it would be good to look at your images, which is why you could totally post on the forum, because that's an awesome question.
Yeah, okay, I will.
Yeah. There's many different answers to that too.
Yes.
There's different ways you can do it.
Thank you very much.
Yeah, no worries. Are there any other questions?
Yes. After segmenting your image, what's the next step to analyze the image, i.e. measuring length, area of your... Yeah,
so that really depends on your biological question. So that's kind of what I was hinting at with all those, that's what you kind of have to ask yourself before you start, right? Before you even start prepping your samples, you have to say, what is it that I need to measure? And then adapting your sample prep. So getting the right markers and things to specifically answer that question, because you don't want to just take a picture and be like, well, what information can I find from this whole process, right? So there's many different things you can measure, but it really depends on your biological question. You can measure whatever you want, right? But not necessarily, but you can measure a lot of things, but you really need to ask yourself that before you even start. So there's lots of things you can measure. So you can measure the length of objects, the area, the intensities within your ROIs, circularity of those objects. You can do shape, things. And we're going to talk more about that sort of thing in the next workshop. It'll be a little bit more practical, because we're actually going to segment and count nuclei and do a few different measurements and things, and then automatically do it with scripting. But yeah, again, it really depends on your biological question. So for me, that's like the first thing I ask people. It's like, what do you want to measure? So
what about colocalization? Colocalization.
about it? About
I
when
mean,
would
in
you
this, if I would like to do colocalization first, when I do all the first steps that you have just described, can I do colocalization or not afterwards to check for?
Yeah. So at least like the plugin that I introduced, you can do regions of interest. So if there's only certain
Yes.
regions you want to do measurements, you can apply them to ROIs. Yes.
Okay.
And just do colocalization measures within that region. So you can apply a mask or do it within individual ROIs. Yeah.
Okay.
Yeah, and again, next week we'll go through scripting and a more practical, and like we'll actually have code that you guys can batch process, and there will be code that we go through that you can actually take and use and modify if you need. So today was just really about a quick intro into the software itself. But again, if you have any questions, you can definitely contact me directly, which is fine, or ask on the forum. There's a whole host of people that are ready to help you guys. So that's what we're there for.
Awesome. Are there any more questions?
I hope it was helpful.
Yeah, I think it was very helpful.
It's harder to tell. Like, I miss being in front of people and, workshops where I can actually like go up to your computer and be like, click on this, but so I hope it was helpful, and we'll see you all next week then, I guess.
Thank you.
Yeah.